Hello. What I'm here to talk to you guys about today are different tools that we can use to study small bio, right? What's happening at the cellular level, what's happening at the subcellular level. We've already used some of these in the lab. We've already investigated some of these in the lab. For example, we took a look at our cheek cells and we used methylene blue in order to identify specifically where the nuclei were. And what you guys found in your pre-lab was that methylene blue was attracted to specific features of those nucleic acids inside the nucleus, and that's why the nucleus was that nice, beautiful dark blue. What you might want to do in a laboratory situation is find different components of the cell. So we have different dyes that are attracted to lipids, we have different dyes attracted to starches, different dyes attracted to proteins. So what we can use are particular dyes based on what we are investigating in that particular moment. You also might want to investigate what's happening to a specific protein or a specific lipid. In that case, things get a little trickier. So we're going to have to delve a little bit deeper into these fields of histology and cytology that we've been using throughout the year. Histology is the study of tissues, and cytology is the study of cells. For both of those, what we're going to do are use microscopes to take a look into this very, very small world around us. So when we're looking for a particular protein, there's a few different techniques that you can use. One of them, and one of the ones that I think is terribly interesting, is to insert a piece of DNA into the gene you're interested in. As you guys know, when a protein is generated, the information that it originally encoded that protein is stored in the DNA. So what you do as a molecular biologist, if you want to find out where does that protein go, or where is it being produced, or find it in any way, what you're going to do is you're going to take a model organism, like Saccharomyces cerevisiae or Mus musculus, like we talked about in class. You can take the gene you're interested in and pop in just a little bit of extra information. That little bit of extra information will be translated along with the gene you're interested in. So that instead of just having the protein, you're going to have the protein plus a little bit of extra on the end. That little bit of extra is something that you'll be able to see. For example, one of the most famous tags is GFP, green fluorescent protein. If you tag something with GFP, what that means is you went to your model organism, you found the gene you were interested in, on one side of that gene you popped in the information encoding GFP, so that when that gene is read, the transcript contains information for the protein and for GFP. And then when that transcript is read, you're going to end up translating it into a protein with a little bit of extra, and that little bit of extra is the green fluorescent protein. So that instead of having just your regular protein, you've got a protein plus a glowing green tag attached to it. What happens because it's glowing is that you can look for it in the microscope, and you can actually find it in particular tissues. You can find which tissues express it more based on how much glowing you see. You can also even go down to a more microscopic level and take a look at where it's traveling within a single cell. GFP is not the only tag. We now also have, for example, a red fluorescent protein tag that you can pop on there. GFP is quite famous, originally came from jellyfish, and it's been put into everything from flies to yeast to rabbits to monkeys to pigs. So it's been quite frequently used. However, it is a fairly large tag as far as that goes, right? If you want to tell it how to glow, you're going to need quite a few amino acids. One of the downfalls to using GFP as a tag is the fact that since it's attached to a protein, that protein might become destabilized by having that GFP hanging off of it. So what might happen is that the protein unfolds, and instead of doing its job, and instead of going to normal places where you'd expect it to be doing its job, it might be degraded instead. If a protein unfolds, it's going to go through something we call the unfolded protein response. Alright, what happens in the unfolded protein response is that the cell recognizes this protein is broken, it's not folded correctly, something's wrong, so it tries to recycle that protein. In order to recycle a protein through that unfolded protein response, we call it the UPR, you're going to have to identify it as broken, and the way a cell does that is by attaching an address tag, which is ubiquitin. Ubiquitin is another small protein. It's a post-translational modifier, which we've discussed at length in class. When you stick a post-translational modifier onto a protein, it can do a series of things. For ubiquitin, in the case of the UPR, what it does is it says, take this protein and degrade it, it is not working. And so what you're going to do with that protein, instead of allowing it free reign of the cell, is you're going to target it to a cellular sort of recycling plant we call the proteasome. And the proteasome will take care of the job of chopping up each of those amino acids inside of that protein, so that instead of having this unfolded disaster floating in your cells, you're going to get amino acids back out, which you can then recycle and reuse to build new proteins. Because of this problem with GFP, what a lot of labs are doing is they're using other tags. For example, there's an HA tag, and that is reasonably short. The shorter the tag, the less chance you have of destabilizing the protein it's attached to. But you're still able to observe where these tags are. The way you observe them is a little trickier. It's a lot easier to find something that's glowing at you than it is to find something you need to fish around for a little bit more. So here we have our cell. Inside our cell we have the nucleus. Inside the nucleus we have a piece of DNA. In this DNA, colored in red, you can see our gene of interest. Colored in green, you can see the bit of DNA that's been added by whatever molecular biologist is working with this cell. That bit is our GFP tag. So that when this gene gets copied over, transcribed into RNA, you're going to transcribe the piece that contains the coding region for the gene and the bit for the tag. As you guys know, that's not the only thing that you copy over. You also copy over an untranslated region at either end. You also have a 5' cap and a 3' poly A tail on that mRNA. Then it goes through splicing to get to its finished form, but the key thing is that you are going to keep this whole piece together. You're going to keep the gene and the GFP tag together. Then you're going to export this out of the nucleus. This piece of mRNA, once it's in the nucleus, is going to be read by our good friend the ribosome. As you recall, the ribosome has two subunits, a big one and a small one. The mRNA is going to sit in between. As this happens, you're going to pull down tRNAs. Those tRNAs are attached to amino acids. As that happens, you're going to start to assemble a long chain of amino acids based on what this ribosome reads in the mRNA. Ultimately, what the ribosome translates from the mRNA is going to be our protein. Let's give our protein some alpha helices, maybe a beta sheet, some more alpha, some what on a structure region. And then at the end, once it's finished reading that gene, what it's also going to end up reading is our GFP tag. Alright, and the GFP is going to cause this to glow, so that when we take a look at this cell, what we're going to see is that glowing GFP. So you'll actually be able to see where this particular protein is because it's literally going to be fluorescing. Unfortunately, as you know, if you add pieces onto a protein, it can destabilize that protein. So instead of having all of those beautiful alpha helices and that beta sheet, what might happen is that it unfolds into a disaster. Right? Now let's connect it all. There we go. So you'll still have your GFP, maybe not, I don't know. Alright? So you're going to have the same stuff, just going to be wildly unfolded. In your cell, if you have things that are unfolded, it can gum up the works for everything else. And so what your cell uses is something called the unfolded protein response, or the UPR. The UPR works by recognizing unfolded proteins, tagging them. The UPR uses its own tag. That tag says, degrade me, I'm broken. Alright? The tag that the unfolded protein response uses is a post-translational modifier. Now, we've talked pretty extensively about post-translational modifiers. What these things do is they get attached to a protein after the fact. And when they're attached to the protein, they can change where it's localized, meaning where it's found inside the cell. It can change what it's doing. It can change a whole bunch of things. The tag that the UPR uses is called ubiquitin. Let me add some ubiquitin here in blue. And typically it adds a few of them, right? Once you add that ubiquitin onto your unfolded protein, what happens is that the cell sees that as an address, sending it for degradation. So now we've got our ubiquitin tag. We're going to go for degradation to something we call the proteasome. The proteasome is basically a big recycling plant. When the unfolded protein gets sent there, what happens is that it ends up going through this recycling plant, and as it goes through that recycling plant, it gets chopped up into little pieces. All of the amino acids that previously made up that protein now are recycled and able to be reused by different ribosomes. So when you have a broken protein, when you have something that's not working, what you do is you attach ubiquitin to it, send it to the proteasome, and degrade it. Alright, we want to be able to use those amino acids for something that actually legitimately works, and not for something that might cause problems within the cell. So that is one of the hazards of using a GFP tag. People still do it because, as I said, it is very, very handy to see a glowing reminder of where your proteins are. And it can give us some very, very good information. However, this is definitely one of the problems that people have to keep in mind when they are using this particular tag. And when they're using any tag at all, in fact. So let's say we wanted to try another technique for figuring out where particular proteins were located. Are they found in cells in dish A, cells in dish B, both? What you would do is take your cells, grow them for a little while so that you have enough of a sample to work with, then you would kill them all and isolate or purify the protein that was in them. So what you can see here, here's our cell. What we did was we smashed it up, we broke the membrane up, we pulled apart all of the pieces inside here. We did all of this at reasonably cold temperatures so that the proteases that were inside the cell weren't given an opportunity to degrade the proteins we were interested in. Additionally, we did all of this lysis, all of this breaking open of the cell in a lysis buffer that had additional protective components that would help prevent that type of degradation from occurring. Once we have the purified protein, you can see I've got a little tube here that should be holding our protein. There we go, there's our protein. Once we have our protein, what we're going to want to do is separate it so that we can figure out exactly where the proteins we're interested in are located. So the technique I would use to do that is called SDS-PAGE. Sodium dodecyl sulfate polyacrylamide gel electrophoresis. And all of that basically sounds very fancy, but what it means, this SDS part, SDS is a chemical that will coat proteins. And it'll coat them in a negative charge. So if you had a protein like this, it would give the outside a nice strong negative charge. The reason we might be interested in something like that is because once you have a charge, we can use an electrical current to help separate you from other components of a mixture. What's going to happen in the gel, this polyacrylamide gel that we make, it's sort of got the consistency of less thick than Jell-O, but not quite a liquid. It's basically a gel where we can put the protein into the top, run an electrical current through it, and because our proteins have been coated in SDS, they will travel along with the current through that gel. Now, this is sort of important. The smaller the proteins are, the easier it is for them to go between the pieces of the gel. What ends up happening is that the small ones drift through this very easily, and the large ones get sort of stuck up near the top. In order to figure out exactly what size protein we've isolated, we're going to include not just our sample of interest. Here we go, we can label that well sample. We're also going to include something we call a ladder. Now, a ladder is a standard. What you have in that ladder are proteins of known sizes, so that you'll have bands, you'll have lines that appear in this gel that correspond to known weights, known molecular weights. So, for example, if our ladder is in green, and we end up with a pattern like this, and the sample we're running, I'll draw that in red. Typically the dye you use in this is blue, but we'll use red for the sake of our illustration. If you end up with a piece of a band right here, a band right here, and a band right here, you would be able to say that the protein you've isolated up here is about the same molecular weight as that part of the ladder. The protein you've got right here is about the same molecular weight as that part of the ladder. If this is 10 kilodaltons, and you know it's 10 kilodaltons, then this protein must also be about that mass. Do you remember our good friend P53? It runs at about 53 kilodaltons. That's how it got its name. So let's review the process SDS-PAGE uses. You take your cells and you lyse them in a lysis buffer in the cold. And all of that is to help prevent the degradation of the proteins of interest. You then purify the protein from the rest of your cell lysate. So you want to get it separated from the lipids and the DNA and the RNA that are inside the cell. Then you coat your protein sample in SDS, and you put it into the top of a gel. This area we call a well. It's just a small little area that will allow you to pipette in your sample. Alright? Whenever you run a sample, you're also going to want to run a well that contains a ladder with proteins of known particular molecular weights so that you can estimate how big the proteins in your sample are. If you want to compare a few samples, you would run them side by side. So for example, if I thought these two samples were similar and I ran them, I'd be able to say, well, they both contain a protein of about the same size, but my second sample is missing this low molecular weight protein. One of the important things that people frequently do if they run a protein gel in this way, is they want to make sure that they loaded about the same amount of protein in both cases. And so what they do then is they soak this gel in something we call Kumasi Brilliant Blue, and then they wash it off just a little bit. The brilliant blue will stay attached to the proteins. So you'll be able to see that the brightness of the proteins is about the same in both wells, and then you can say, you know what, I was about right, we loaded about the right amount. There's other things that you can do with this gel. Once you've separated the proteins based on their molecular mass, as we've done here, we can go fishing for particular proteins. Now it is incredibly hard to go fishing for particular proteins inside that gel. That gel, as I said before, is sort of jello-like consistency. In order to go fishing for the proteins of interest, what we're going to have to do is move the proteins from that gel onto a thin membrane. The way we do that is again with electrical current. If I were to place a membrane over the top of this gel, and I were to put a buffer all around it and were to run an electrical current through it, the proteins would be moved from the gel onto the membrane. So you'd be left with a piece of sort of slimy feeling paper, which we call a membrane. You'd be able to see the ladder. What did I put my ladder as? Something like this. Close enough. You'd be able to see your ladder. But you wouldn't see anything in the wells where, or in the areas where you ran your sample. In order to visualize what's there, we are going to use a technique known as Western blotting. What we do for Western blotting is we literally go fishing for particular proteins. We're going to use something we call antibodies. We're going to try and attach them to the proteins we're interested in. So let's say I get the antibody attached to my protein of interest. What I would do next is try and cause it to glow. Again with the glowing, it's very easy to see things when they're glowing. Once I have this glowing, I'll be able to see where on the membrane it's located. In order to record that so that I can use that information later, I will expose this membrane to photo paper. Just like you would if you were developing old school photography paper. So let's review the steps of Western blotting. It's very much like SDS-PAGE in this first part, right? You're going to grow or obtain the cells you want. You're going to isolate the protein. Remember, to isolate the protein, we need to lyse those cells in a lysis buffer. We want to keep it cold to prevent reactions. We don't want proteases to chop up our proteins of interest. We want them as big as they are really in real life in the cells. Then we're going to coat them in SDS so that they're coated in a negative charge. We're going to run them on a gel. We're going to load them on the gel. Then we're going to run an electrical current through the gel so that the large proteins stay towards the top and the small ones are pulled pretty far to the bottom of the gel. Then, in the case of Western blotting, we are not done. In the case of Western blotting, what we're going to do next is transfer the proteins to a membrane. That is crucial because the next thing we're going to do is go fishing for our proteins of interest using antibodies. So if you have a protein you're interested in, an antibody would attach to it, and then we're going to go on to the next step where we're going to do a chemical reaction that causes those antibodies to glow. In order to figure out what molecular weights have these now glowing proteins, these proteins of interest, we're going to expose a photoreactive paper to the membrane. This is the same type of paper you would use if you were doing old-fashioned photography. Once you expose that photo paper, you're going to develop it, again, just like you would with old-fashioned photography. You go into a dark room, you put it through some developer, and you'll get out at the other end something that looks like this. You're going to have the piece of the photo paper that was exposed to the membrane showing dark bands wherever the glowing protein was located. And it'll glow more intensely if you have more protein there. So it'll help you compare a few different samples. So let's review what we've talked about today. When you're studying small bio, we can use microscopy. It's a very, very powerful tool to look at life on the tissue level, which we call histology, or at the cellular level, which we call cytology. Very often they're clumped together. We've talked about the need to use dyes in order to obtain a location for particular cellular components. The dye we've used in class is methylene blue, but you've also seen some others in some of our prepared slides. And dyes can attach to pretty much anything. We can find some way to make things colorful. And it's easy for our eyes to observe where particular colors are located, which made the discovery of green fluorescent protein such a big deal, right? Because if you can tag a particular protein, let's say you're interested in P53, if you attach green fluorescent protein to it, you can actually observe where it's localized, where it's located, based on where you see green glow inside the cell. One of the dangers of using these types of tags is that you might not be able to make as strong a conclusion as you think you might. You might say, ah yes, it's glowing green in the nucleus, therefore my protein is in the nucleus. Or you might say it's glowing green inside the cytoplasm, therefore my protein is in the cytoplasm. And that's what tags are good for. But when you have a large tag, especially one that causes something to glow, you might end up unfolding the protein you're interested in, and instead of seeing where it's really located, you might send it through the unfolded protein response. What happens there is that your unfolded protein becomes degraded. There's a few types of microscopy we spent some time on this year: scanning electron microscopy, transmission electron microscopy. For those two types of microscopes, your samples cannot be alive because they must be done in a vacuum. Tags like GFP aren't terribly successful with electron microscopes because what happens in the electron microscope is that instead of observing light, you're using a beam of electrons. And since you can't see that glow that GFP gives your protein, you're not going to be able to tell where your GFP tagged protein is using these tools. You're only going to be able to observe where GFP is located using light microscopy. Other things we've talked about as far as techniques you can use on small bio include SDS-PAGE. SDS is a chemical that will coat our proteins of interest with a negative charge and allow us to separate them on a polyacrylamide gel using an electrical charge, basically, so that the small proteins end up at the bottom of the gel, the large proteins lag behind. In order to get to the point where we can separate them all, we use the following steps. We lyse the cells we're interested in, coat the proteins in SDS, load our proteins onto the gel. Along with our proteins of interest, we're going to add a ladder of known molecular weight, so that we know the molecular weights of the proteins we've separated. If we wanted to visualize where the proteins ended up in SDS-PAGE, what we can do is add to the gel something called Kumasi brilliant blue, which will cause the areas that have the protein to turn blue. If you were interested, instead of just seeing generally where all of the proteins were, if you were interested instead to find out where a particular protein was located, we can take the gel from SDS-PAGE and use it for our next technique, which is called Western blotting. In Western blotting, what you do is you take the gel, here we go, you take the gel and you put a membrane on top of it. You then run an electrical current through the gel so that the proteins end up transferred to the membrane. Once you have your proteins on the membrane, you're going to go fishing for your protein of interest using antibodies. You're going to use a chemical reaction to make the areas with the antibodies glow. You're going to expose that membrane to photo paper, and then you're going to develop that photo paper. So you will be left with a black and white image where there's dark bands wherever that protein was located on your original gel. We have a bunch more tools that can be used, but we will save those for another day.